So, hi everyone, thank you for coming. I'm Brooke Gilbert. I'm a senior at Pinecrest School, and I conducted research on red tide and utilizing a native seagrass, Gracilaria Parvispora, in mitigating red tide blooms at the Urban Farming Institute in Oakland Park, Florida. So a little bit of background information on red tide. Red tide is when the organism Karenia brevis blooms to populations of higher than 1,000 cells per milliliter. And this bloom occurs due to excess nitrate and phosphate levels in the environment, which can occur due to runoff of products such as untreated sewage, agricultural runoff such as fertilizers and livestock waste. And it affects areas along the Gulf Coast as well as Florida, and more specifically, the Mississippi River area region, as well as areas near the St. Lucie and Caloosahatchee rivers in Florida, because these are areas where a lot of agricultural runoff are dumped into the ocean. And Karenia brevis is such a problem because when it blooms, it takes all the oxygen out of the ocean, and this can lead to a lot of fish die-offs. Vegetation can't grow when there's no oxygen in the water. And it also releases a brevetoxin that leads to fish die-offs as well, and can also lead to human illness when these brevetoxins are inhaled or people eat contaminated shellfish. And currently there's no solutions to this issue, and it's a very big problem in Florida, and it's only been getting worse. And each red tide bloom, it's super expensive. It's estimated to cost $51 million per each red tide bloom. And that's extremely expensive, and that includes the issues it causes for the tourism industry, the health costs due to people getting ill, as well as the beach cleanup costs because of all the dead fish along the ocean. So a little further on Karenia brevis, it's considered a pretty smart organism. It can actually hunt for its food, and this is a little bit rare, considering most organisms just die off when there is no available nutrients. Karenia brevis actually swims to its nutrients, so it's a very versatile organism, and it never completely dies off. There's always a background concentration of at least 1,000 cells per milliliter in the oceans, so there's always an opportunity for a red tide bloom. And it has a wide range of food sources. Not only is it photosynthetic, it also derives its nutrients, as I said before, from nitrate and phosphate, as well as dead fish and animals and the sediment of the ocean floor. And these are some micrographs I actually took using the microscope at the Urban Farming Institute right here. So those are them just swimming around there. These are some additional micrographs. So one thing I wanted to point out here, the micrograph at the top left is a picture of the Karenia brevis after it was fed. And you can actually see the bits of mysis shrimp inside of the cells. However, the picture on the bottom left, the red outlining is due to the brevetoxin release. And it's green because it is reproducing photosynthetically. It's deriving its nutrients photosynthetically. So that Karenia brevis cell is starved for nutrients. And that's where we see the red coloration in the ocean during the end cycle of a red tide bloom. So the other organism I used in this experiment was Gracilaria Parvispora, and so a little bit of background on that. It is a red algae. It has antibacterial effects, and it has very high sequestration rates of nitrate and phosphate. And it's native throughout the Atlantic coasts as well as South Asia. And the reason why I chose this organism was because I saw a paper that outlined the experiment of these Chinese scientists who utilized Gracilaria Parvispora to clean up their polluted waters in the South China Sea, and it had a drastic effect. Wildlife started to return. There was much lower nitrate and phosphate concentrations in their waterways, so I decided to use this organism for that reason. And The reason why we don't see any effects of the Gracilaria Parvispora in Florida is because most of their populations have been wiped out or just decreased due to development and pollution and things like that. So we haven't been able to record the effects Gracilaria could have had on decreasing or mitigating red tide blooms for those reasons. So after I pitched my idea to the Urban Farming Institute, we started construction on the lab, and so I wanted to kind of break that down because it was a little bit important to the experiment. What we have is a system that pumps fresh water from the sump, which is the larger kind of conglomerate of water, and it pumps it into the water chiller, and then from there it goes into the other larger white tanks. And so the smaller saltwater tanks sit inside a water bath of fresh water to maintain the temperature throughout. And we were able to use this system since it's so flexible and more economical than other methods. We're able to use this system for the entire experiment, as well as culturing the Karenia brevis and culturing other seaweeds. So that's just another picture to kind of visualize it besides that graph. So those are the larger water tanks, as I said before, and the sump is on the floor. That pumps it into the water chiller, as you can see up there. So I had to visit Mote Marine Lab to get my Karenia brevis sample, because even though it's in the ocean, we needed to get a specific strain, and there was no way to verify if I was receiving the right strain if I just took a sample from the ocean. So I went to go get the Wilson strain, which is the most prevalent red tide Karenia brevis strain that blooms during red tide. And so I went to Mote Marine Lab, and these are some of the pictures that I took while I was there. In the top right corner, there is a picture of their culture laboratory. They have massive stores of Karenia brevis cells at all different concentrations. It was super interesting to have the opportunity to go there. So after that, we began to culture the Karenia brevis. And so we were culturing it in jars, and we then moved on to tanks, and we were developing a nutrition method, as well as trying to perfect our method of keeping the right conditions for Karenia brevis to grow. And we would keep track of our cell counts using a hemocytometer and prophy tests and pH to maintain the saltwater conditions. So preliminary research I had to do before I actually started my research project was finding out the correct nutritions for Karenia brevis. And so we would use Chaeto grow, nitrate and phosphate concentrate, and mysis shrimp cubes. And we developed a formula for the UFI lab, about 2 grams of mysis shrimp per 5 gallons of brevis culture. So that is a very high, that's providing the Karenia brevis with a lot of nutrients so that we could get the culture to really high cell counts. And during this culture process, we ended up observing a bloom cycle of brevis, which was pretty interesting. We saw high population blooms and then really low population die-offs. So to the left is what the Karenia brevis looks like under the microscope, and then to the right is what it looked like in the tank. So as you can see in the microscope, this is a micrograph of healthy brevis cells under the hemocytometer, and this is kind of a micrograph and comparing it to the tank of the brevis reaching high population levels or approaching the bloom phase of the cycle. So we see the Karenia brevis kind of accumulated on the sides of the tank, and that just shows a really, really high concentration of Karenia brevis in that tank there. And then we also observed massive die-offs. So on the left, the brevis cells are clumped together and sort of lysed, and they formed large clusters of dead cells whenever they would die off. And we would see the foaming at the top of the tanks, indicating the brevitoxin release. And Karenia brevis releases its brevitoxins when it's dying off, not while it's raising in population. So we could confirm that this was a brevetoxin release. And the green color in the tank would indicate the photosynthetic activity of the Karenia brevis, because now that it's starved for nutrients, it needs to make up for this by acquiring its nutrients photosynthetically. So we would observe that in the tanks by the green coloration. So then I got to get started on my actual experiment. And so my hypothesis was that when I introduced the Gracilaria into a red tide simulated environment of Karenia brevis populations higher than 1 million cells per milliliter, Gracilaria Parvispora could outcompete the Karenia brevis for nutrients and therefore decrease Karenia brevis populations and decrease the associated red tide effects. And I ran this experiment using the same mason jars that I had originally cultured the Karenia brevis in. And then I ran 4 trials, and there were 2 control groups and 1 experimental. One control group was just Karenia brevis, the other control group was just Gracilaria parvispora, and then the other was the Gracilaria with the Karenia brevis. And there was a standardized consistent feeding rate. And for data collection, I utilized the hemocytometer to do the cell counts and just monitor the population levels over time. And then I also maintained the tank chemistries by Profi testing and pH. And something else that I did was testing the photosynthetic activity of the Karenia brevis using a spectrometer. But later on, if I run another trial, I'd actually like to do a spectrophotometry, which is a more advanced analysis of the actual composition of the Gracilaria parvispora as well as Karenia brevis to indicate which organisms are sequestering how much of the nitrate phosphate. And so this is a picture during trials, and the results were really astounding. The jars with just Karenia brevis, which are the red and blue, their population levels stayed extremely high the entire trial. So this indicates that because of the high feeding rate, it's sequestering all these nutrients and it's maintaining a very high population level. So this was a very severe red tide bloom in those jars. However, in the jars with the Gracilaria and the Karenia brevis, the Gracilaria was able to decrease the Karenia brevis population level consistently throughout the entire experiment. And actually, in some cases, the Karenia brevis population reached below background concentrations, which was very significant for this experiment. Another thing that I tested was the weight of the Gracilaria parvispora. So the Gracilaria weight with Karenia brevis, which is the red and blue lines in each of these, that indicates there was an increase in weight. However, it wasn't an exponential increase or a huge increase as significant as the jars with just the Gracilaria parvispora, which increased in weight a lot more than the jars with the Karenia brevis. So this just indicates that the Gracilaria was competing with the Karenia brevis for nutrients. However, since it's increasing in weight, we do see that it was able to outcompete the Karenia brevis and gain the nutrients. Not only that it needed to maintain its current structure, but actually was able to build and grow. And this was consistent across all trials, so this was also indicative of the beneficial effects of Gracilaria parvispora. And lastly, I measured the photosynthetic activity of Karenia brevis. So on the left is the photosynthetic activity of Karenia brevis for the test jars that had no Gracilaria introduced to them. And this indicates that there was photosynthetic activity occurring during these trials, which shows us that the population levels had grown so high that they were sequestering all the nutrients in the water and needed to make up for the nutrient deficit that they were experiencing by acquiring their nutrients photosynthetically. And to the right, the graph shows the photosynthetic activity of Karenia brevis in the test jars without the Gracilaria. And since there is no photosynthetic activity, it indicates that the Karenia brevis was acquiring all the nutrients that it needed with the nutrition inside of the jars, and that was also significant as well. So this was just some of the media coverage that we received, and this helped us promote the experiment. And I also just wanted to make some acknowledgments. First to Ms. Liz Dutra at the University of Miami, as well as NOAA, Dr. Dennis Hanziak and Dr. Philip Wills at Harbor Branch, Mr. Cody Cole, who helped me acquire my red tide Karenia brevis sample, Ms. Gordon-Air and Ms. Gandon, who have mentored me at Pinecrest, the University of Connecticut, who provided me with critical background research that I needed to conduct this experiment, Dr. Katie Hendrickson at Nova Southeastern University, Ms. Emily Timothy, who helps us with our chemistries in the lab, and Daniel, who comes in and volunteers and helps us maintain the lab as well. Mr. Jeff Manchester, who helped us design the lab and who's also encouraged me throughout this entire experiment. And then lastly, this experiment would not have been possible without Mr. John Albee. He was critical in this experiment, and just a big thank you to him as well. So these were the citations, and if anyone has any questions.
What was your highest cell count that you had during the trial?
So he just asked what was my highest cell count. My highest cell count that I ever recorded, I believe, was about almost 1 billion cells. It was very close. I think almost 750 million to 800 million cells, which is a very severe red tide bloom. Okay.
How long did this take you?
So she just asked how long did this take me. This took me, I started working on the actual experiment in the UFI during May, and it took me until about December of this year. We were designing the lab, and I was working with Mr. John for a while before that. However, actual testing started in about May, and I worked over the summer in the UFI to get the results and everything.
So you said that the native population of the Gracilaria was depleted because of development. How much of it is needed to mitigate the red tide? Like how much is there to be out there?
So we used about 20, she just asked how much would we need to implement into the natural environment to have the same beneficial effect for red tide. We used about 20 grams of Gracilaria Parvispora during the trials, and that was able to mitigate about 1 million cells per milliliter. So if you use that formula, depending on the red tide bloom and the severity of it, you need to implement about 20 grams of Gracilaria Parvispora per 1 million cells per milliliter.
Is there a way to detect when you think a red tide bloom is going to occur so that you could introduce the Gracilaria before the red tide actually blooms?
Is there a way to detect when a red tide bloom is going to occur? Not necessarily. There are seasons where red tide blooms are very severe, and that's because of the agricultural seasons, the runoff that occurs due to that. But you can't specifically detect when a red tide bloom is going to occur unless you know of runoff or sewage runoff or agricultural runoff that's been let into the environment. You could predict that a red tide bloom could occur.
Has there been any correlation as far as farming and runoff with the seasons so that you could actually maybe get a good start I introducing something that was prohibited in college?
So she just asked if there's a correlation between the agricultural seasons and the red tide blooms. There definitely is. There's actually been a lot of legislation that has been proposed to have the big sugarcane farmers, because they're the largest culprits specifically for Florida. So there's been a lot of legislation proposed to have the big sugarcane farmers have less runoff or control their runoff, because they know that their runoff is the biggest culprit in the red tide blooms, but none of it has been successful. But yeah, they do know that it's due to specifically the big sugarcane farmers in Florida. He just asked if this is common among Gulf Coast states or if it's just Florida. This is common. It's actually more severe in the Gulf Coast due to all the agricultural areas along the Mississippi River. Most of the runoff actually flows down the Mississippi River and is let out into the Gulf Coast. And the largest dead zones are recorded in the Gulf Coast. There's hundreds of square miles where there's no fish or wildlife because the red tide blooms have been so severe. And it does affect areas, Texas, Louisiana, places like that along the coast of that the red tide blooms are just very bad over there. Yeah.
Is it exclusive to saltwater environments or affected freshwater environments?
Karenia brevis is exclusive to saltwater environments. However, there are different blue-green algae that can affect freshwater environments and have similar red tide effects as the Karenia brevis does.
So is this affecting our manatees here?
She just asked if this affects manatees and things like that. It can affect manatees. Usually the first organisms to die off are the small fish and things like that, but in a severe red tide bloom, it could be possible that they could kill off big mammals like that, manatees, yes.
You said you can't predict when a bloom is going to start, but if there were testing of the concentration where you could see a buildup, it's starting to build up, or does it happen too fast for monitoring to help out that?
It does happen pretty quick. The Karenia brevis can sequester nutrients almost overnight. I saw sometimes when I was culturing, populations would go from 500,000 to 1 million in one day. So it grows very, very quickly. And so if they were to predict it, if they got lucky enough to see it coming, they would have to take action right away, because red tide would occur pretty quickly after that.
So if they took action right away, how long would it take to decrease the population of the Karenia brevis?
So during my trials, here I'll pull it up, the Karenia brevis population levels, those are the green and purple bars. It decreased gradually, so it would just prevent even if it didn't prevent the red tide bloom altogether, it would prevent the severity of the red tide bloom, because even in the test trials without the Gracilaria Parvispora, you see the Karenia brevis population spike, but with the Gracilaria, even if they don't go down immediately, the Karenia brevis doesn't spike. So it would prevent severe red tide blooms, even if it didn't prevent the red tide bloom altogether. Okay, thank you everyone for coming and listening to my presentation.